Welcome, everyone. Today we're going to explore one of the most powerful analytical tools used in modern laboratories, gas chromatography mass spectrometry, or GC-MS. GC-MS is often called the analytical workhorse because it can do something very few techniques can: separate complex mixtures, identify unknown compounds, and quantify them with confidence. By the end of this video, you'll not only understand how GC-MS works step by step, but you'll also understand why it is trusted across pharmaceuticals, forensics, and environmental science. Imagine you're handed a colourless liquid. It smells faintly sweet, but that's all you know. Is it safe? Is it pure? Could it contain trace contaminants that might trigger a safety recall? This is where GC-MS steps in as the detective of analytical science. GC-MS doesn't just separate compounds, it names them, quantifies them, and often tells us where they came from. When decisions impact patient safety, product quality, or legal outcomes, GC-MS is the tool scientists trust. Real-world samples are rarely simple. Pharmaceutical products, environmental samples, and forensic evidence all contain complex mixtures. Gas chromatography alone can separate compounds based on boiling point, but it can't tell you exactly what those compounds are. Mass spectrometry can identify compounds, but only if they enter one at a time. GC-MS solves this by combining both strengths. First, compounds are separated. Then, each compound is identified individually. This combination is what turns GC-MS into a complete analytical solution. Think of GC-MS as a two-stage journey. In the first stage, gas chromatography, compounds separate like runners in a race. Each one exits the column at a different time, known as the retention time. That timing alone is already a valuable clue. In the second stage, mass spectrometry, each compound is broken into fragments, creating a unique pattern that can be matched to known libraries. Simply put, GC tells us when a compound appears, MS tells us who that compound is. Everything begins at the injection port. A small amount of sample is injected into a heated inlet where it is instantly vaporized without decomposing. This vaporization step is critical. An inert carrier gas, usually helium or hydrogen, then sweeps the vaporized sample into the system. The gas doesn't react, it simply transports. Here's a practical insight. Poor injection technique can lead to tailing peaks, split discrimination, or loss of volatile compounds. In GC-MS, good data starts with good injection. Once vaporized, the sample enters the GC column, a long, narrow tube coated with a stationary phase. As compounds travel through the column, they interact differently with this coating. Some stick longer, some boil later, and some pass through quickly. These differences cause compounds to separate in time, producing distinct peaks. Column chemistry and the temperature program play a huge role here. This is why GC method development is often described as both science and art. After separation, compounds exit the GC column and move into the mass spectrometer through a heated transfer line. Temperature control here is critical. Too cold, and compounds condense. Too hot, and they may degrade. Why does this matter? Because the mass spectrometer must receive one compound at a time. This clean handoff is what allows GCMS to produce clear, interpretable spectra and accurate identifications. Inside the mass spectrometer, molecules enter the ion source. The most common method is electron ionization, or EI. High-energy electrons strike the molecules, knocking out electrons and breaking them into fragments. This may sound destructive, but it's intentional. Each compound fragments in a predictable and unique way, creating a molecular fingerprint. That fingerprint is the foundation of compound identification in GCMS. Now that we have ions, the system must sort them. This is done by the mass analyzer, most commonly a quadrupole in GCMS systems. The quadrupole filters ions based on their mass-to-charge ratio, or MZ. Only ions of a specific MZ reach the detector at a given moment. By scanning across a range of MZ values, the instrument builds a mass spectrum, showing ion intensity versus MZ, a unique signature for each compound. The detector measures how many ions arrive at each MZ value. The result is a mass spectrum, which acts like a molecular barcode. This spectrum is compared against reference libraries such as NIST. When the spectrum matches and the retention time aligns, you get high-confidence identification. This reliability is why GCMS is trusted in pharmaceuticals, forensic investigations, environmental testing, and impurity profiling. Here's the key takeaway. GCMS works because separation and identification are synchronized. Every step, from sample preparation to ionization, affects the final result. When you understand the working principle, you're not just running a method, you're interpreting chemistry. GCMS doesn't just answer, "What is this?" It explains why it behaves the way it does. Thank you for your attention.